Welcome back to Introduction to Chemical Engineering. Krishna Chitur. We're going to talk about two products where it's going to be clear to you that biology is very important, and we'll try to make a connection with the engineering of these products, as it were. Again, what we're going to do, look at the product, look at the concepts, some of the ideas, in this case, some of the concepts from biology, and then take a look at what the role the engineer might play or has played in the manufacturing of the product. The product we're going to look at first, we're going to look at two products these next 2 weeks. Well, the first product we're going to be looking at is something called penicillin. It's an antibiotic. I think most people are familiar with it, antibacterial. And what is the role of the engineer in making this antibiotic? Okay. So again, reminder, the enabling science we're talking about now is biology. And we will talk about the relationship of biology to the products, penicillin and insulin, and the role of the engineer. Okay, a little bit of a history with penicillin. I encourage you to read up about it. It is sort of discovered by accident, if you will. It sort of started in 1921 when the scientist noticed that there was a petri dish where the bacteria were growing, and then he dropped something. It turned out some of his nasal secretions. He wanted to study it and take a look. And it turns out that he noticed that wherever the secretions fell on the plate, the bacteria did not grow. So he said, oh, interesting. Maybe there's something in those secretions that prevented the bacteria from growing. But it turned out that that happened to be something called lysozyme, which is a protein that breaks down other proteins. So it's still useful, but not as useful as a drug. And so, but again, it also did not destroy all bacteria. Several years later, the same scientist working in the lab was working in some other petri dishes where bacteria were growing. And he noticed that there was a region in the petri dish where the bacteria were not growing. And this time it was not nasal secretions. So he said, why is the bacteria not growing there? So he thought, well, is it a contaminant of some kind? Was something else in the petri dish secreting some chemical stopping the bacteria from growing? Or what it was? Anyway, to make a long story short, he discovered that there was a molecule in that zone of inhibition, as it is called, that is preventing the bacterium from growing. And that turned out to be penicillin. This was approximately 1928, and it has a structure as you can see here. So what was also interesting and fortuitous, really, to Alexander Fleming was that the bacteria he was growing in that petri dish happened to be sensitive to penicillin, which was being made by another bug, if you will. Now, and that bacterium happened to be gram-positive bacteria. Don't worry about the details, except that if he had been working with gram-negative bacteria, that penicillin effect that he saw, he would not have seen. So in a lot of ways, he was lucky because he was working with gram-positive bacteria, and it was penicillin sensitive. So he discovered. And one of the things that scientists do is discovering something works. While it is wonderful, you need to know how it works. And the reason for that is because it will help us design production methodologies, and also actually be able to improve it if you need to. So scientists are not just asking what, but also how. So today we know a great deal about penicillin, how it works, how it kills bacteria, and so forth. And so for penicillin, for example, and different antibiotics have different ways in which they stop bacteria from growing. And that's also important to know. Penicillin works by stopping the growth of the cell wall, as it is seen over here. So this is a protein that is involved in growing the cell wall, and penicillin binds to it and prevents that protein from working. So simple as that. So history. So Alexander Fleming discovered penicillin, wonder drug. You might think, okay, well, can we use it right away? And if you look at the date, this is 1941, and some of the history I got is from this book, Microbe Hunters. 13 years later, is when the first experiment was actually done on a human who was very sick and with infection. And out of desperation, somebody actually injected this little boy, I think, with Albert Alexander, with penicillin. And he actually got better. But of course, it turns out that they needed more penicillin, and there wasn't enough penicillin, so that actually patient Alexander actually died. And so, but it proved that penicillin could stop infections. That is very important. And so you take a look, 1921, discovery, 1928. 1921 was the lysozyme, 28 was the discovery of penicillin, 1941 is when the first experiment was done. It takes a while, so something exciting in the lab doesn't necessarily translate right away. And there are many, many reasons for that, and certainly one reason is engineering, understanding how to make this in large quantities safely, so on and so forth. So engineering remains a challenge even today. So as I said, lysozyme to penicillin to clinical trials, space of about 20 years. Okay, so what made the production of penicillin important is World War II. And around World War II, at that time, more people were dying on the battlefield of infections than wounds caused by the battle itself. And so people said, is there another way, is there a way to prevent these soldiers from dying? And penicillin came to the rescue. And then the people decided that we need to make large amounts of penicillin and of course package them in a way that we can transport them to the troops and help them. And so the engineering of production of penicillin, purification of penicillin, and transportation of penicillin turned out to be very important, and engineers, chemical engineers in particular, played a very important role in that. And as you can see, the scale up of the penicillin production in 1943, again, don't worry about the units. Say 21 billion units were being produced, and by in a year, it went up 2 orders of magnitude, 1,600 units. So it was so important, and so that was actually made. And here is several important people involved in this process. Alexander Fleming, who discovered it, and then Chain and Flory, who were primarily responsible for scaling up the production. And it's a fascinating story, and I would encourage you to read more about it if you can. And then, of course, we don't use the same strain of Penicillium chrysogenum as it's called today in the lab, so in the factories to make penicillin. But we have discovered other strains, things that are higher producers, better producers, and so forth. We've also, the engineering of this was in not producing penicillin in petri dishes, but in completely different novel ways, where they can make large quantities of them. And that was actually using something called submerged fermentation. It's engineers came up with this idea. And then, of course, the next challenge after production is to purify them. You have a large quantity of water, a dilute solution of penicillin in water. How do you recover it? How do you purify it at prices that are affordable? So that was also an engineering marvel. In fact, the term liquid-liquid extraction was actually used for purification purposes. So it's a fascinating story in production, the engineering of production, the engineering of purification, and identification, and so forth. And so the scale-up was achieved by fermentation and submerged fermentation processes. So the takeaway from this few minutes is you have an observation. We understand a little bit better about the science as to why the chemical and how the chemical worked, and then the engineers came and made the production of this particular chemical, penicillin, in large quantities actually feasible.